Is Eglantine Hector. She is a PhD student at the Marseille Immunology Center in France, where she's in the group of Pierre Millepied. And in her PhD, she's exploring broadly germinal center selection by BCR signaling and TFH-derived cues, as well as selection mechanisms during plasma cell differentiation. And today she's talking about a pipeline for inference of phylogeny and selection pressure in BCR sequences. Yes, looking forward to your talk.
Do you see my screen?
Yes.
Okay, nice. Do you hear me as well?
Yes, everything.
Okay, nice. Okay, so hi everyone. So today I'm going to present the pipeline for BCR sequencing SeQuoIA that I have been implementing during my PhD. Okay, so a bit of background on B cells. So they express immunoglobulins in the form of a B cell receptor or as antibodies. In either case, immunoglobulin is made of a constant part that mediates effector functions or transduces BCR signaling. And the variable part is involved in antigen binding. So these receptors are highly diverse and they can bind antigens with high specificity and affinity. And all these features, diversity, specificity, and affinity are acquired throughout B cell development and maturation. So it all starts in the bone marrow, where the initial diversity is generated through VDG recombination. And upon infection or vaccination, naive B cells can either readily take part in the extracellular response or undergo further maturation steps in germinal centers. So within the dark zone of germinal centers, B cells can further diversify their receptors through the somatic hypermutation process. So point mutations are introduced in the coding sequence encoding for the variable part of the B cell receptors. So obviously it can alter the binding properties. And these binding properties are going to be tested in the light zone, where a follicular dendritic cell presents the antigen to the maturing B cell. And they thereby gather several cues in the light zone, which will affect their fate. So they can either undergo apoptosis or be positively selected and be licensed to go back into the dark zone. And alternatively, they can differentiate into effector B cells, so plasma cells or memory B cells, and be involved in future responses. So this germinal center reaction and the accelerated evolution with mutation and selection falls within the Darwinian theory that is illustrated here. So we have this initial variation within a population. Individuals compete with one another, and it's the survival of the fittest. And some traits confer a competitive advantage and are more likely to be passed on to the next generation. So this is the same for B cells. We have this repertoire diversity, and some receptors are more likely to be passed on to the next lymphocyte generation. And during affinity maturation, we know that higher affinity cells expand within the population. But how exactly binding properties translate into a competitive advantage is not fully understood. And these are the questions I tried to tackle during my PhD. So I wanted to uncover the link between binding properties of the B cell receptor and its carrier fitness, and also try to uncover new mechanisms for germinal center selection. So to answer these questions, I have been analyzing single-cell RNA-seq data. So we have access to the transcriptome and also to the sequence encoding for the B cell receptor. With the former, we can identify B cell subsets, activation state, or signaling transduction events. And with the second, thanks to mutation accumulation in the germinal centers, we can trace back B cell dynamics and evolution. And so this is a classical BCR sequencing pipeline. Usually it starts with sequence assembly. Sequences are aligned onto a genome so as to infer the germline. So the sequence before mutation accumulation. VDJ segments are annotated, and cells are grouped into clonotypes and should correspond to a single lineage. And finally, intraclonal evolution can be represented as a phylogenetic tree. Okay, so I have implemented SeQuoIA to harness this kind of integrative single-cell data and link BCR features to a transcriptome. We propose some improvements of the steps that I have presented in previous slides, including phylogeny reconstruction. But most importantly, we introduced new methods to infer selection based on mutation patterns. So this is an overview of the pipeline. So for the pre-processing steps, we just applied incantation, and now I'm going to dive into the phylogeny part. So there is already plenty of tools for phylogeny reconstruction that are custom for BCR sequencing data. For practical reasons and based on these two benchmarking studies, we chose to base ourselves on GC3, which is a maximum parsimony method, which means that the number of mutations is optimized and minimized. So we have for each clone a collection of heavy chain sequences and light chain sequences. We apply the parsimony method, but it rarely leads to a single solution. So we have this collection of trees that are equally parsimonious, which is called the parsimony forest. What GC3 does is to select one tree within the forest based on node size distribution, with the premise that expanded nodes are more likely to be divided and to have a descendants. Also, it uses somatic mutation patterns and selects trees that best fit mutation hotspots and cold spots. So we chose not to restrict ourselves to the parsimony forest. We also explore a larger space of solutions with a print and regraph algorithm. And also we introduce new criteria to choose the best tree within the solution space in a way that accommodates current knowledge of B-cell biology. For instance, we want intermediate sequences to be productive because we know that unproductive mutations lead to B-cell death, and we don't want that. Also, we check that the isotype order is respected. So for those who come from the biology field, you probably know that this is an irreversible process and that it occurs in a certain order, starting from IgM to IgGs and so on. So we want, for instance, IgMs to be upstream of the tree axis here. And most importantly, since we have trees for heavy chain and for the light chain, we have this concordance score that checks that both trees, each node have concordance, consistent lineages in both trees. And also we check that there are no aberrant or reverse mutations and so on. So we checked whether SeQuoIA successfully optimized the criteria that we have introduced. So this is the case. In the right panel, what you see, we compared the concordance scores between SeQuoIA and GC3. So it just corresponds to a tree pair within a clonotype, and we see that the trees selected with SeQuoIA have more consistent lineages than the one chosen by GC3. And here, in this red rectangle here, you see that it's on trees that corresponds to birth trees that are consistent. And SeQuoIA further optimizes lineage overlap between both genes. Despite our efforts, there were residual aberrant trees with SeQuoIA, which didn't reach a solution. And so we filter them out before further analysis. So we only want to keep the green part here, the only concordant trees. So this is a manual steps, and we let the user perform it. And there can be some permissiveness to rescue some large clones of interest, for instance. We hypothesize that these aberrant trees were stemmed from issues in preprocessing steps that could propagate in subsequent steps of the pipeline, such as phylogeny reconstruction. And so we proposed one improvement for clonotype assignation. So we split sequences by donors before hierarchical clustering. And indeed, we improved tree accuracy by introducing this improvement. So all this suggests that phylogeny tree reconstruction quality is dependent on preprocessing and data quality altogether. Okay, and now that we have reconstructed our trees, the next step of the pipeline uses these selected trees to infer selection pressure. How does it work? So here is a schematic representation of a phylogeny tree. We use all the intermediate parents nodes here, sequences, as an input for a somatic hypermutation model. So we introduce mutations in silico according to this S5F model, which accounts for AD bias. And we end up with a set of virtual sequences that we compare to the experimental ones, which are derived from the same parent. And we interpret the divergence as being the effect of selection. And the tree is really key here because we can distinguish instantaneous from cumulative selection. And also, we're convinced that it improves modeling accuracy by restraining the time window over which mutations are modeled. So how do we measure this divergence? We use this metric from information theory, which compares two distributions. And the distribution are the probabilities of being mutated for each codon. And since we compare sequences with different mutation loads and different backgrounds, we normalize this scale divergence with entropy. And this is our selection score. And before applying our pipeline, we wanted to test the robustness of our selection score. And to do that, we cross SeQuoIA output with gene expression data, and if available, we also compare them with experimentally characterized immunoglobulin properties. So first quality control was to check whether selection imprint was detected in the right cell subsets. So, as I said in the introduction, mutations are accumulated in the dark zone and selected in the light zone. So the B cells that are transiting from the dark zone to the light zone should have unselected mutations, whereas the few ones that pass through light zone selective processes should have increased in the selection imprint when they recycle back to the dark zone. And this is what we see here. So we plot selection scores across germinal centers B cell subsets. So here are just steroid clusters that we have annotated. And so we see a nice increase in selection imprint from the moment they enter the light zone to the moment they exit the light zone. So our first quality control was validated. Another quality control was a correlation with MYC, which is selection marker. And we see that our selection score nicely correlates with MYC expressions, so which was expected as well. And finally, in one of our datasets, we had access to affinity measurements. So we wanted to know whether our selection score were meaningful and reflected immunoglobulin affinity. So the lower the KD, the higher the affinity, and we see that indeed our selection scores nicely correlate with affinity. So I'm just showing a few examples, but we've performed this kind of validation in both human and murine datasets, so we were quite happy with that. I hope that I have convinced you that our selection scores are reliable. And now I'm going to present a few applications of SeQuoIA implementation. So it can be used in any dataset with ongoing mutation accumulation. So it can be to study fundamental germinal center biology, vaccination monitoring, tertiary lymphoid organ reactions in tumor microenvironments, and also in germinal center-derived pathologies such as lymphoma or autoimmune diseases. The main downside of SeQuoIA is that it requires the tree for selection pressure inference, and it only works on datasets with clonally expanded sets. So it requires some throughput and a certain amount of data, and also it can't work on blood samples because they're not clonally expanded. Okay, so now I'm going to show one example. So we applied SeQuoIA to our SARS-CoV-2 vaccination cohort, and we had nice results about selection at germinal center entry and exits. So again, we plotted a selection score distribution across cell clusters. And we see that there is some persiveness in the first mutations that are accumulated upon germinal center entry. And we see that this selection imprints when B cells are fully fledged germinal center phenotype. Another interesting result was that mutations were accumulated during the extrafollicular response, and they seem to be subject to selection. Afterwards, we wanted to study the germinal reaction in this dataset, as well as germinal center-derived plasma cells. So since we're in human here, we don't have reporter genes or lineage tracing methods. So we use this little trick here with our BCR sequencing data. So we subsetted only cells in the trees that have completed a few cycles over the germinal center reactions. And here are the results. So again, we see this increase in selection imprint in the light zone. And also, we saw that early plasma cell precursors were not selected, but there was an increase in selection imprints in late plasma cell precursors. And we hypothesized that there is a selection checkpoint during plasma cell differentiation. Okay, so we applied SeQuoIA to several datasets, and we proposed this model of germinal center reaction. So we propose that there is some selection during the extrafollicular responses. That germinal center entry is permissive, permissive over the first mutations that are accumulated. We have this selection step in the light zone that was already known. We have intermediate selection scores for memory B cells, and this increase in selection over plasma cell differentiation axis. Afterwards, we wanted to dig a bit deeper into the mechanisms underlying light zone selection. And to do that, we screen all the genes that were expressed in the light zone, and we correlate them with SeQuoIA selection scores. We selected positively correlated genes, so 600, and we ran a gene ontology. And here are the results. So, of course, cells with high selection scores tend to be more activated. They populate some metabolic programs and seem to reshuffle their cytoskeleton. We propose novel signaling pathways to be involved in light zone selection, such as interference. And most importantly, it seems that light zone B cells, selected light zone B cells, were conferred with a pro-survival advantage, with a protection against intrinsic apoptosis, but also against the extrinsic pathway, suggesting that negative selection could operate through active elimination of light zone B cells, and that they would not just die by neglect, as it is sometimes suggested. And the last application I want to show you was measurements of selection scores over time in a murine response or in the same human cohort. And we see that instantaneous selection increases over the course of BCR response, suggesting that there are some feedback mechanisms that make selection more and more stringent over time. And this is it. Well, thank you for bearing with me. And I would like to thank my supervisor, Pierre Mipier, and also I'm grateful to all these people here that helped me a lot to handle datasets or their code and integrate, for instance, GC3 into the pipeline, which was a challenge for me. And also the AIRR-C organizers, of course. And I'm happy to take questions.
Great. Thank you very much. You can probably stop sharing.
Yeah, sure.
I'll see you. Yeah. Thank you very much. Are there questions already from the audience? Otherwise, I will ask a question. Oh, there's Artem. Yes, please.
Hi, thank you for a beautiful presentation. May I ask you one question about the reconstruction of the trees, about forcing the isotypes to be in the biological order? And it also relates to the second question of the size of the trees, which we want to see. So I'm sure that that's a correct approach to enforce that you have biological order of the isotopes, because in those trees you could actually have sampling, and you could have some of the more mutated IgMs, which happen to be from a different branch, happening in your data, but you'll force them to be upstream of something else, right? So it could be in principle that you just, well, because of the sampling, it happened to be that you have some IgMs which are more mutated and are downstream on this tree. Yeah, and I'll probably proceed with the second question. And the second question is, it feels like with this approach, it could only be correct if you have pretty large trees. What is your estimate for the minimum size of the tree? Because like I feel like this selection metric would make no sense if you have a tree, I don't know, of 4 members or even 5 members, because these probabilities will be so noisy that it literally will ruin the whole idea.
Okay, so I'll start with the first question. So actually, so the class switch criteria comes last. So we apply the criteria in a certain order. So it's not the main one. So we pre-select trees before applying this criteria. So for instance, productivity and concordance comes first. So it's more important than class switching that comes last. And also we know that class switching is supposed to occur before germinal center entry. So I think that the IgM part should be divergent from the IgM, I guess. The IgG, sorry. And what I'm penalizing is that whether we have IgM being the mother of IgG, but if they are in separate branches, there is no penalty. But we don't filter them out. We just rank the remaining trees. And there is a figure actually in the paper, and it has a little impact because it's one of our last criteria. And for the second question, so it was about tree size. Okay, yeah. It's true that in many cases the trees are trivial, so it's just the ancestor and the daughters. So there are no flowers in the tree. But yeah, even in small trees, we can make some corrections. Maybe can I share my screen again or not? Okay. Yeah, so maybe I can show this little example here. So yeah, we see that even in a small tree, we can still select for best solution. So here we see that this one is paired to this one. Obviously, this can't be because yeah, it should be linked to the ancestor, and this is not the case. And SeQuoIA chooses this one that is more likely than this one, for instance. I hope that makes sense.
Yeah, thank you. May I ask a couple more questions?
Yeah, sure.
We have 5 minutes, but yes, yeah.
Yeah. That's just very interesting and right on my lane. One quick question about this. Have you compared to any of the implantation lineage reconstruction tree tools like Dowser, or like something which you have in Dowser, or most importantly, I think for me is have you looked at comparison with IGFIML? Because I feel like this is the most
Yes.
advanced tree reconstruction now, but well, very slow, but still probably the best one.
So I had issues with IGF-female actually. So I tried it indeed. So according to our criteria, SeQuoIA performed best. But yeah, it's actually the new week format that you use that is not the same as GC3. So you can't just switch the tree reconstruction as it can't be chosen by the user because they're not compatible. So this was a challenge for me. I had to adapt the new week format to be readable by AIR packages, and this was very difficult.
I see. Well, I think if you contact Kenneth Herhen, who's the author of this, he could help you with this. He's very responsive and always helps with stuff. So yeah, I encourage you to do this. It will be very interesting to see the comparison.
Okay. Thank you.
Habib?
Yeah, thank you. Thanks for the nice presentation. I have a very kind of almost touching the territory of the previous. I've only used Dowser before to construct phylogenetic trees, and there is like a place or like a step where you need to define like a sequence similarity threshold in order to say that these sequences fall under the same clonal family. Is there something similar in the pipeline that you presented that kind of defines that these sequences coming from the same clonal family?
Yeah. So maybe I'll share my screen again. No. Sorry, maybe slow. Okay. So indeed we have yeah. So there is this clone type assignation that comes before, and it's just a threshold, a single threshold, and it's usually 80%. It can be altered, changed by the user, but in practice, when it's more stringent, well, you lose data and then statistical power for selection score analysis. So we haven't tried many clone type assignment methods, just playing with the threshold and this donor-based approach. I don't know if I answered your question. Okay.
But Artem has another question.
Yeah,
Artem.
Artem.
Yeah, it's actually been shown that this kind of approach and 80% similarity is very loose for this. So you usually with this you get huge clones which are usually not really clonally related, but just happen to have similar rearrangement, and this happens with the shorter CDR3 lengths typically. So there are alternative tools which overcome this. One of them is actually from one of your colleagues, Thierry Mora and Alexandra Valzac.
You mean Igor?
No, the Igor is the generation thing. There is a thing called Hillary, I think, and they tackled this problem beautifully there. I would say I like this tool the most.
Okay, I kind of realized this clone type issue in the end of my thesis, so it's a bit late now. But yeah, indeed, I wish I could had time to test several methods.
Yeah, I think we have 0 minutes left, but just very briefly, because we are the AIR community, I think I need to ask these questions. So, is your pipeline AIR compliant, and also how fast does it run? How compute intensive?
Okay. So yes, supposed to be AIR compliant. So the output is just a data frame. What SeQuoIA does is to add the columns for selection scores to the AIR formats. It also adds balance with tree information, such as who is the parent, who is the daughter, the nucleic string, and so on. There are also other outputs that are not independently of formats, such as tree quality, which are in separate data frames. And your second question was about
speed.
The time or something.
Yeah, speed. Yeah.
Okay.
Speed.
Unfortunately, it's quite long actually.
Hmm.
So can be in a small data set can be one day, but in the human COVID, it was 3 weeks. So very long. Yeah. So,
that's long.
So the bottlenecks are clone-type passing grouping, which is quite long actually. GC3 was long, and mostly tree selection was as long as GC3. So it doubles the GC3 computation times.
Great. I think if there are no more urgent questions, thank you very much for the beautiful talk also.
Thank you.
We'll be back in February for another seminar. And thanks all for your question, and have a nice day, everyone. Thank you. Bye-bye.